Alright, this is going to be Chapter 11, the cardiovascular system and the blood, Part 1. Introduction to this, the cardiovascular system pretty much has a pumps and pipes and the ability to carry the blood to the body. There's both a mechanical aspect to this and a lot of biological systems are involved, and any of these can malfunction, causing fluid loss and or pump failure. Very simply, we have three things that make up blood pressure. They're in kind of a triangle here. Contractile force strength, which would be the pump or the heart rate, and then peripheral vascular resistance, and then the fluid is pretty much encompassed by those three properties. If the heart's not pumping hard enough or fast enough, or the vessels are the wrong size, then you will not have adequate blood pressure. And if there's not enough fluid for those three things to work with, again, you will not have adequate blood pressure. So we'll talk about this more whenever we hit shock and things like that in the chapters of the paramedic curriculum. Functions of the blood. Transportation of dissolved gases, nutrients, and hormones and waste. So pretty much this is the piping system for all of our hormones and anything that would regulate other areas and organs in the body. Regulation of pH, which is essentially acid. So the blood regulates the acid levels of the body. Restriction of fluid losses at the injury sites. So example of this, if you have an arm here and it becomes severed, in this area here, the vessels that were coming out will actually contract and suck back up inside. So this will help in fluid loss if you've actually lacerated a vessel. Defense against toxins and pathogens. If you have things that don't belong, like bacteria running around the system, the body easily will mount a defense against it. And stabilization of the body temperature. It's kind of like a radiator and hoses that in the wintertime. So not only does it cool the system by taking heat away from the outside or dispersing it over a larger area, it also controls sebaceous glands, sweat glands, constriction, a whole multitude of things that would be in the regulation of body temperature. Composition of the blood collection and analysis in this section. Plasma contains dissolved proteins, slightly denser than water, and I'm going to read through these and then we're going to get to a slide with some pictures on it and I'll kind of break it down. Formed elements, blood cells and cell fragments, which are pretty much platelets. Platelets give us the ability to essentially clot, or they're emergency patches, if you will. If you're bleeding and blood's flowing out of the area, the platelets assist in stopping the bleeding process. In the formed elements, we have red blood cells, and so we essentially have two sections. We have plasma, which is clear stuff, and then we have formed elements. And another thing here before we go too far, if you have a blood sample and you get a red top or tiger top tube, which is a multicolored red top tube, it'll have something called serum separator in the bottom of it. So they put blood in here and they essentially spin it down, and after it spins down, what you're going to get is we run tests on a red top that have to do with the electrolytes and the plasma and things like that. So this serum separator is lighter than the red blood cells but heavier than the plasma. So what you'll get is you'll get the plasma up here after it's been spun down, the serum separator or that gunk that's in the bottom of the tube here, and then all the red blood cells here. And this is a actual vacutainer. So whenever they get done with this, they'll take their sample from this section here for this specific tube. Now each tube has a different type of additive in it. So the different colored tubes have different chemical additives. So composition of the blood, we have two parts that we're concerned with here, one being the plasma and the other one being the formed elements. And in the formed elements section, we have essentially red blood cells, white blood cells, and then platelets that are running around in the sample. And all of these would be down here in the sample collection. Red blood cells are erythrocytes, or red cells. They transport oxygen and carbon dioxide, way important. White blood cells are leukocytes, function as a part of the body's defense mechanism, and these things will attack or have macrophagic action on foreign invaders. And we'll talk about macrophage and phagocytosis here in the second part. And what this essentially does is this means that the white blood cells will attempt, or they will eat, the foreign invaders. So they surround their cellular, their external part of the cell around the foreign invader, and they essentially envelop the foreign invader inside of them, and then the stuff inside of their cellular area pretty much does the number on them, takes them down, uses chemical warfare. So we'll talk about that too. That happens in sepsis, that this becomes problematic. So last but not least, we have platelets, and they have they're pretty much enclosed cytoplasm, or the fluid inside the cells, and they contain enzymes and factors important in blood clotting. So these guys are, if you lacerate a vessel, this is what's going to assist your body in bridging a gap so that the bleeding process will stop. And then last but not least, we're talking about this entire section here, which is whole blood. Contains both plasma and the formed elements. Blood collection and analysis. We have two types of punctures that we see most of the time in the emergency room. One of these punctures is a venipuncture, and the other one is an arterial puncture. So one of them comes from the deoxygenated side, and the other one comes from the oxygenated side. Puncture of a vein for venous sampling evaluates plasma contents, whole blood samples. Examples of this are going to be a complete blood count, any kind of electrolytes like sodium, potassium, CO2, chloride, any of those samples will be drawn off of the venipuncture or the plasma sample. And then we can do ammonia levels off of these as well, all from a venipuncture. There are tons and tons. This is just a small cross section of tests that can actually run on venipuncture. However, these essentially, the venipuncture is the most common test done, and there are literally hundreds of tests that you can run on a venous sample. And then we have an arterial puncture, which is kind of isolated. An arterial puncture is an arterial stick, so they're going to actually stick, the arteries are a little deeper, and they pretty much assess gas exchange, and this is going to be the amount of O2 and CO2 that are actually present in the arterial sample. So this gives them an idea of how much gases that you're carrying around your body on the oxygenated side. They also do a pH on it, a base equivalent, and a bicarb standardly on these. And what this tells them is how much oxygen is in that sample, how much CO2 is in that sample, if there's enough bicarbonate in the system to act as a buffer, since the blood pretty much adjusts pH. The base equivalent, I meant to do an E there, the base equivalent is the differentiation between the pH and the bicarbonate level. And then last but not least, the pH, and this tells us the acidity level. So normal acid levels for a human body are going to be 7.35 to 7.45. And if you haven't got this number in your head now, it would be a good thing to do so. All right, so this is the venipuncture side, and then in this sample here, which is a sample of whole blood, we have two parts. And the two parts, we have plasma, which accounts for about 55% of it, and then we have the formed elements, which is, these are the cells, and this counts for about 45% of it. Now, in the plasma section, and this is going up here, we have plasma proteins, and albumin is the most abundant plasma protein. We have globulins, they transport ions, hormones, lipids, have an immune function. An immunoglobulin is an antibody. So Examples of antibodies would be IgE, IgD. These are going to go out through the body and initiate the immune system since they're traveling around in the blood continuously. This also brings into question, and we'll talk about it more in the immune chapter, to how the body initiates the immune system. So we have an antigen-antibody reaction that generally occurs, and from that we get an immune response. Fibrinogen, essential components of clotting. This is the pre-phase. Fibrinogen will actually form fibrin, and this is what allows essentially the bridge or the matrix that your blood clotting will stick to if you have a large hole in a vessel. And what this does is this allows the platelets something to stick to. Fibrin polymers are formed from thrombin, which is the actual thrombosis. So all of these will assist in the stopping of the bleeding. Regulatory proteins, enzymes, proenzymes, and then hormones also in this section, which is here. And then other solutes, and this is a very small portion, which is only like 1%. But we have electrolytes, organic nutrients, and organic waste. Now, the blood also carries around the wastes of other organs to get them out to the various sections that they need to go to. Things that would filter the blood would catch these wastes, like the liver and the kidneys. Electrolytes, all of our central electrolytes are actually in the blood supply and can be assessed from there as well. And then other nutrients, ATP production, growth, and maintenance of cells, fatty acids, glucose, amino acids. We have amino acids running around. We have blood glucose levels that are essentially in the plasma. As far as the formed elements, now we're talking about the lower half here. The formed elements are the cellular bodies themselves. So we're going to start at the bottom here and kind of work our way up. We have red blood cells, which are about 99.9% of this formed element. So whenever we see something like a hematocrit essentially gives us a percentage of the formed elements. Now in that, 99.9% of them are going to be red blood cells. The white blood cells and the platelets form about 0.1% of the sample. But these are essential. If you don't have enough platelets, you could bleed to death. These are what stop the actual bleeding process in the acute setting. White blood cells, these are your defense against foreign invaders and pathogens in your body. And we're going to talk about the various types in part 2. Neutrophils or polymorphonucleated white blood cells, basophils, lymphocytes, and then monocytes. All of these will be talked about in part 2 and what they actually do. But again, we have 2 parts to the venipuncture. We have plasma and then the formed elements. And the formed elements are the cellular bodies. The plasma would be the clear fluid. Plasma and plasma proteins, and this would be from the top section of that figure 11.1. Plasma makes up approximately 55% of the volume of whole blood. Water accounts for about 92% of this 55%. In the plasma proteins, we have albumin, most abundant protein, major contributor to the osmotic pressure. And what this is going to do is this is going to allow us to move fluid appropriately from the extracellular environment to the intravascular space and so on. Globulins, which are antibodies and transport proteins, about 35% of this is in the plasma sample. And then we have antibodies or immunoglobulins, and these things pretty much attack foreign invaders and pathogens. All immunoglobulins, let's put it this way, I started to say that incorrect. All antibodies are immunoglobulins. However, all immunoglobulins are not antibodies. And whenever we get to the immune system, we'll talk more specifically about the antibodies that we're wanting to look at and their function in the body. Transport proteins, they bind small ions, hormones, or compounds. An example of this is going to be thyroid hormones. This is going to be from your endocrine chapter. And then we have fibrinogen, changes to form fibrin, and this is the basic framework for blood clotting. And then we have just the serum. The fluid left over after the clotting proteins is serum. Chemistries are run off essentially of the serum. So after all of these proteins are accounted for, what's left over essentially is where the electrolytes are located at, and this would be considered the blood serum sample. Formed elements, and this is going to be of the cellular bodies, the lower half of figure 11.1. The production of formed elements, red blood cells, sickle cell disease, rapid blood tests, emergency transfusions, white blood cells, hemolytic disease of the newborn, testing for blood compatibility, and then blood transfusion reactions. The most abundant formed elements are the red blood cells and white blood cells. Platelets are essentially non-cellular packets of cytoplasm, and they have a very functional role in the clotting process. So again, the formed elements mostly are made up of red blood cells and white blood cells, and then a very small portion is made up of platelets. Red blood cells carry the gases in your body, oxygen and CO2. White blood cells are big in defense, and then the platelets are functional in the clotting process. Pardon me. The production of formed elements. Now, we're going to eventually look at a picture that will make all of this make sense. But until then, we essentially have a process that we're going to talk through. And then also we are going to have a lot of terms in this first section. When we get to the picture, we'll talk about all those terms again, but we'll kind of show you and break it down for you at that point. All right, so my apologies, I didn't even start at that. The production of formed elements. Now, these are all the cellular bodies that we're talking about here. Produced through a process called hemiopoiesis. And embryonic cells develop in the first 8 weeks and become hemocytoblasts. They divide to produce myeloid stem cells and lymphoid stem cells. Liver and spleen take turn in the development. Bone marrow takes over as the primary site of blood development. So this happens in the first 8 weeks of life, and after that, once your bones start developing, your bone marrow is where your red blood cells or the formed elements are produced from. Red blood cells. The red blood cells contain a pigment on them that's very high in iron called hemoglobin, and this binds and transports oxygen and carbon dioxide. A hemocrit is a percentage of the whole blood volume occupied by the cellular elements or the formed elements. And in adult males, the formed elements of the blood sample, of the whole blood sample, should be about 40 to 54%. In adult females, about 37 to 47%. Now, talking about the structure of the red blood cell, during the formation, they lose their organelles. And if you'll remember back from a previous chapter, the organelles are all the little parts that are inside the cell. So the red blood cell has no organelles inside of it whatsoever. Without the mitochondria, they obtain their energy only through anaerobic metabolism. So these things are under low power all the time. Aerobic metabolism, you make an abundance of ATP. Under anaerobic metabolism, you generally only form 2 ATP molecules. So these things run on low power, and they have no organelles. Hemoglobin structure and function. Hemoglobin is responsible for cells' ability to transport oxygen and carbon dioxide, and this is going to be essential. If you do not have the red blood cells, then you do not have the hemoglobin present. Without the hemoglobin, you do not carry oxygen or carbon dioxide back from a cellular environment. And this is a real sample here of red blood cells on a stained slide. This is an artist or computer drawing, so you kind of see what they look like. And none of these, if you'll look in here, none of these have any kind of mitochondria, nucleus, or anything like that. They do not have any organelles. Fortunate for us, they do have hemoglobin, which will allow them to carry oxygen and CO2. Sickle cell disease. Sickle cell disease is caused from a sickling of the red blood cells. In normal hemoglobin, there are 2 sets of chains, 2 alpha and 2 beta chains of hemoglobin. In sickle cell disease, the beta cells are replaced by something called S hemoglobin. Now what this does is under oxygenation, things are fine. So the red blood cell looks like it normally should, which kind of looks like a donut. When deoxygenation occurs, or this cell, the hemoglobin gives up the oxygen in its cellular environment, then the S hemoglobin kind of does its thing. And what it does is it will cause a sickling or a curving of that red blood cell. And what this causes is we get something called hypoxia from this. And this will increase the viscosity of the blood. It does this in two forms. One, the body keeps thinking that it needs more red blood cells. Two, these things can actually become pretty sharp and they'll stick on cellular environments. They obstruct small vessels, they hemolyze rapidly in about 10 to 20 days, which also is problematic because we normally keep them around for about 120 days. So these things don't have much of a lifespan, and the patient is essentially hypoxic most of the time, or will have clogging problems. Now, any place to where we would have small vessels, like the lungs, kidneys, liver, any distal part, we can get swelling or viscosity changes in the blood or obstruction of the small vessels. This is a normal red blood cell. This is figure 11.3 in your book. And then this is a sickle cell. So sickle cell causes these sharp points, and this is where these will stick to areas that they're really not supposed to stick to. And their lifespan is 10 to 20 days instead of 120. RBC lifespan and circulation. Hemoglobin urea. What this is whenever there's a bunch of red blood cells that are destroyed or their byproducts in the urine. Urine with a reddish brown tint caused from a large amount of red blood cell breakdown. Hemolyze, which brings up another term, ruptures and becomes devoured by phagocytes. So whenever a red blood cell finds something that it doesn't like, let's just use that as an example, it can do something called hemolyze. And what that means is that it can break down into like little small pieces or destroy itself essentially. And whenever hemolization or rupture of that red blood cell occurs, now you have all of these parts that are floating through the system, and they can clog up vessels. They can actually excite clotting. You can get pulmonary embolus in your lungs. You can clog up your liver and cause portal hypertension. You can go into kidney failure, all from the same pathology that we're talking about here. An example of hemolization would occur whenever a person has type A blood and you gave them accidentally type B blood. This could cause a hemolytic reaction. All the red blood cells start to burst. The patient would become hypoxic. All the organs that filter blood would become clogged up. Multiple problems could occur. Bilirubin, another term, yellowish breakdown product of normal hem. Catabolism, which is breakdown of a red blood cell in its normal lifespan. Hem is a part of hemoglobin. Jaundice, yellowing brown to the skin with high amounts of bilirubin free floating. So we get bilirubin from the breakdown of red blood cells. The liver has a big gigantic part in this. So people that go into liver failure can have high bilirubin and become very jaundiced from this. Transferrin, transporting proteins of the blood, transport iron to transports iron to the end is absorbed by the developing red blood cells. Okay, so transport iron and is absorbed by the developing red blood cells. Excessive transferins are removed by the liver and spleen where iron is stored. And we're almost to the picture, so bear with me here. Recycling RBCs, or red blood cells. Red blood cells live about 120 days, and they're broken down by macrophages, or the white blood cells. And then the iron is reutilized. Red blood cells are broken into biliverdin, which makes essentially bilirubin. Bilirubin is converted by the liver and expelled in the large intestine with the help of bile and eliminated in the feces. Iron is reutilized by the help of transferrin, red marrow, for formation of new red blood cells. Okay, so first picture here. All right, so we're going to start talking about this guy about right here. He's had a 120-day lifespan, and he hemolyzes. So the macrophage breaks all of this down and tries to reutilize as many parts as it can. The hem is sucked up by the biliverdin, which is utilized by the bilirubin, and it goes to the liver for elimination in the large intestine and the kidney. The iron is reutilized and transported by transferrin out of the macrophage and into the red bone marrow, where we get red blood cell formation, and then the new release of a red blood cell into the circulation. And we get about 10% hemolysis from these. So whenever we kick these things out kind of quick, we only get about 90% of the product. Any of these that hemolyze will go through the same process. Back out, iron, bilirubin, liver, kidneys, and so on. This process occurs every second of the day in your body. Red blood cell formation. Lots of terms here. We essentially have one more page before we actually hit the picture that I was talking about, and it makes more sense than actually the terms do. So bear with me through the terms, and then we'll talk about it on the picture. Hemocytoblasts, and originating cells of white blood cells and red blood cells. So both our white blood cells and our red blood cells come from these things called hemocytoblasts. Development divisions of hemocytoblasts make myeloid stem cells, and we'll talk about myeloid tissue and myeloid stem cells here in just a second. Erythropoiesis, which is red blood cell formation. So anytime we see something called EPO hormone, erythropoietic hormone, or erythropoiesis, this means new formation of red blood cells. Myeloid tissue, which is essentially red bone marrow, and this is where the actual blood cells come from, white blood cells and red blood cells. Myeloid stem cells, daughter cells of the hemocytoblasts, and since these guys started all off, these are going to be the daughter cells from them, and variations can occur on these myeloid stem cells. Erythroblasts are immature red blood cells that actively synthesize hemoglobin, and these things will actually become red blood cells or kick out or produce red blood cells. In this process, they lose their organelles. Reticulocyte, next phase of maturity of the red blood cells, makes a reticulocyte. They circulate for about 24 hours, and then they become a mature red blood cell. In this 24-hour period is where we see some of them actually hemolyze. And this is the picture I was talking about. So all of these cells down here at the bottom, let's start at the bottom, and this means that our red blood cells, our platelets, and any type of white blood cell all are made originally from one type of cell that comes from our bone marrow called a hemocytoblast. Now, from this, whenever they daughter off or make a new cell or create a new cell, so this guy has made a new cell, and this one is scheduled to be a red blood cell. So it comes under the effects of erythropoietic hormone from the kidneys. This now becomes a progenitor cell, or it has the ability to know what it wants to do in life. It makes a blast cell, or a proerythroblast. Once it is an erythroblast, this is scheduled, it's committed at this point. It's going to make reticulocytes 24 hours after circulation. It becomes a red blood cell. Progenitor cells also here. This can change into a megakaryocyte, And what this does is, see all of these little packets that it has here? These will actually form and become the platelets. Another progenitor cell, and this comes under the effects of colony stimulating factor. This makes a myelioblast instead of proerythroblast, and then this can actually form various types of white blood cells off of this one type of cell. And these are band cells, which are immature white blood cells. This one is going to be a basophil, an eosinophil, and then a neutrophil. Now, this right here is an important thing to remember. This is an immature white blood cell. This guy cannot handle a full-blown attack of a pathogen. The neutrophil, however, can. And if we'll notice here, do you see all of this area that's in this thing? This is called a band. And the reason that it's called a band is because all of these nucleuses are still banded together. This is how we would identify this as a immature white blood cell. This one here, this neutrophil, has multiple individual nucleuses. So whenever they become mature, then we have another name for them. They're called a polymorphonucleated white blood cell, or a neutrophil. Now, why that's important? Poly meaning many, nucleuses. Now what this is this is a mature white blood cell that can interact and attack an invading pathogen. From the progenitor cell, monoblast, different type of colony stimulating factor, promonocyte, and then it'll actually make a monocyte, which is a different type of white blood cell. Lymphoid stem cells, also derived from the hemoblast. This is the only one that's outside of this pathway off of the myeloid stem. And this is a lymphoid stem cell. This will make a lymphoblast, which will make a pro-lymphocyte, which is an immature lymphocyte, and then a mature lymphocyte. Big, gigantic macrophagic actions. In part 2, we'll talk about the white blood cells. But right now, we're going to probably stick with this side of the map. All right, regulation of erythropoiesis. Erythropoietin, also called erythropoiesis-stimulating hormone, appears in the peripheral tissues, especially the kidneys, after low oxygen concentration, and it is released when you're anemic or have decreased blood flow to the kidneys, oxygen in the lung declines, respiratory surface of the lungs are damaged. So anytime that they see hypoxia in the system, they pretty much release EPO so that you can make more red blood cells. EPO performs two tasks. It stimulates increased cell division of erythroblasts and in the stem cells that produce erythroblasts. It speeds maturation of the red blood cells. Under EPO, red blood cell formation increases tenfold. So this EPO hormone supercharges the whole let's produce red blood cells. And then this is a term, please realize this, that low states of oxygen in the tissues is called hypoxia. All right, so this is figure 11.6 in your book, and we're going to start it right here, that the kidneys discover that the tissue oxygen levels decline. So the kidneys will kick out EPO, and this is an endocrine hormone, erythropoietic hormone. And what this is going to do is off of the stem cells, the blank cells in the bone marrow, it's going to increase their meiotic rate, which means that they're going to kick out more erythroblasts, and these things will actually make more reticulocytes, which are going to be immature red blood cells. The EPO also says that, hey, we can't just have you all sitting around and playing Xbox forever. We're going to increase your maturation. So it matures them faster, which will get them out into the system faster. And this is going to increase the number of red blood cells, which improves the oxygen content of the blood. It has more carrying capacity, and tissue oxygen levels rise. And hopefully this will correct it. If not, we'll get this again. Clinical notes, rapid blood tests, and these are going to be if someone comes in with signs and symptoms of shock, they're going to do rapid blood tests. And we have about a 4-hour window, give or take, and I'll explain this here in just a second. An H&H blood test, which is an hemocrit. Remember, 99.9% of the formed elements are made of red blood cells. And then we also look at the hemoglobin content. So the H&H stands for hemocrit and hemoglobin. The hemocrit is a standard look at the formed elements in the blood, but 99.9% of them are the red blood cells. And then we also look for the amount of hemoglobin that's present in the body. Now, if they're both low, then something has to be done. Either the person is put on rest and low stress so that they can make their own EPO, or we give them a blood transfusion. In the acute setting, most of the time, we will lean towards the blood transfusion if the patient will accept it due to their religious beliefs. A 4-hour window, let's explain this. If a person is bleeding out actively and they essentially draw a blood sample, the hemocrit and hemoglobin, or the H&H, may be normal. 4 hours after the event occurs and we get the appropriate amount of fluid back on board, because the fluid will concentrate. If when we take another H&H, the percentage of the red blood cells may have decreased. Whenever you're looking at a percentage of the red blood cells, you're looking at the percentage in that sample. So if the person is dehydrated, or if the person is suffering from hypovolemia or low volumes of fluid, then you may not get an accurate H&H test. So about 4 hours is what is looked at. So after the event occurs and you stabilize the situation with fluid, then another sample should be drawn about 4 hours after things are corrected. What this is going to do is this is going to show them a more accurate cross-reference of the blood sample at that point. RBC and blood types. Antigens, substances, most often proteins that can trigger an immune response. The surface of a red blood cell contains 50 different kinds of surface antigens. And this is going to lead us to the ABO system, our blood typing system. Type A has antigen A only. Type B has antigen B only. Type AB, both antigen A and antigen B. And we're going to talk about this, but again, the picture is a lot easier to take a look at. RBC and blood types continued. Type O has neither A or B antigen. This whole Rh positive stuff, we need to talk about this here for just a second. Rh positive is another antigen that is present on the blood. So whenever we talk about blood type, there's 3 types. There's there's 4 types. There's A, B, AB, and O. And then most of the people know their blood type by I'm O positive or O negative or A negative or A positive. The positive or negative is determining or stating that they have an Rh positive or an Rh negative. And this is another antigen found on the red blood cell. Agglutinogens, all access pass on the red blood cell, and this is immune system recognition. So the agglutinogens pretty much tell they have like an all access pass, and what I mean by that is that this red blood cell belongs to Roy Smith. So my red blood cells that are running around my system essentially have a hall pass that says they can do that. If they did not have that hall pass on there, then my body would attack them. The white blood cells would attack them, the other red blood cells would attack them and clump up. So this is why that blood typing is essential. We should always cross-type and match somebody. In a pinch, we can give them something called O negative. And the reason, and I'll point it out here, type O has neither A or B antigen. So this type O blood has no presenting antigen or presenting hall pass. He's like the UN. I can go anywhere. Now the Rh positive or Rh negative, the best type to give them is going to be O negative, because the negative means that they have no Rh antigen on the red blood cell. Next term here, agglutinins and plasma will attack antigens on foreign red blood cells. All right, red blood cells and blood types, antibodies and cross reactions, and we're going to take a look at 11.7. That's going to be the figure that we're going to look at. Next slide. Cross reactions, which means we have an antigen-antibody reaction. Agglutinations, and an agglutination is clumping up after an antigen-antibody reaction. So an example of this, if you have type B blood, one red blood cell that's type B and one red blood cell that's type A, and you put them in a petri dish together, they're going to clump together and initiate something called an antigen-antibody reaction. And then we're going to talk about a term called compatible. And compatible means that there's no clumping seen on the blood typing assessment. All right, so this is figure 11.7 in your book. So type A has a surface antigen for type A. So this all access pass pretty much says that I am type A. I am Roy Smith in type A. And they essentially have the antibodies present for type B blood. So if you put type B blood into a person with type A, we would have an antigen antibody reaction, which would excite the immune system. And just to make note, this is what happens when we excite the immune system. We get something called hemolysis, or the blood cells essentially burst. So type B blood, going to this one that we just talked about, type A, has an all access pass that says I am B. And they have the antibodies for type A. So again, if we, I'm going to erase this here just so that we keep clear on it. If we put in type A blood into a person with type B, we would have this, an antigen antibody reaction. All right, type AB blood. They have surface antigens for both A blood and B blood. So they have an all access pass for A and B. Consider them like a hall pass. They have no antibodies for either A or B. Type O blood. They have no antigens on their surface at all. So this guy's hall pass, he can go into anybody. They do have antibodies, anti-A and anti-B. So if you put, he can only take type O. If you put type B in, isolated, or type A in, there would be one of these. All right, with that said, we have surface antigens on these guys right here, and then we have opposing antibodies. They clump together or agglutination or clumping, and hemolysis occurs. So the red blood cell itself kind of bursts and breaks down. Type A can only take type A. Type B can only take type B, or I should say, or type O. Type AB can only take type AB, or I'm sorry, type AB can take type A, type B, or type O. And type O can only take type O. All right. Clinical notes, emergency transfusions, 450 milliliters. O negative is the universal donor. The reason it's the universal donor, it has no presenting antigens. We will not get an antigen-antibody reaction because it doesn't know what it wants to do with its life. and it has no RH factor. So it's indifferent there too. AB is referred to as the universal recipient. So an AB person has no antibodies present, down below if you'll remember from that figure, has both A and B antigens on the surface, so we can tape A, B, or O. I'm sorry, that should be an A. And that's why it's called the universal recipient. And O is the universal donor because of this. No antigens on its surface. This concludes part 1. If you have any questions, feel free to contact me, 405-219-7613, or you can reach me at smithr@imsa.net. Thank you.